Hey guys, so in this lecture we're going to talk about chapter 8. So we are going to go more into detail about the genetics and dabble a little bit in the biotechnology side of bacteria. So we're going to go a lot more into detail of bacterial genomes and actually how do they replicate their DNA, how do they make their protein synthesis. A lot of it's very similar to us, but there's also some differences, and we're going to talk about both. So let's go ahead and get started. Oopsie, there we go. So the bacterial genome, as we know, it's not linear like how it is in a eukaryotic cell. So they organize it very differently. They have some bacteria will have just the standard genome, which is if you were to like stretch it out, it's one big loop. And then sometimes they'll even have they'll have extra genomic material, which is considered as a plasmid, and that is also circular as well. So the difference between bacteria and eukaryote is that their genome in bacteria and prokaryotes it is circular, whereas eukaryotic is more linear based. And then with this, I'm not going to expect you to know actual like how many bases are in their genome. It was more or less just a table just to show you that yes, it's a bacteria, but different bacteria, they will have different counts of genome. So like sometimes their genome will be bigger, sometimes it will be smaller, just depends on the species. That was the idea of this slide. So next we're going to talk about actually like what makes DNA relatively unique. So DNA, it's in all living cells and it's considered the blueprint of the cell. So it's consisted of chromosomes that is double-stranded. So DNA is double-stranded, whereas RNA is single-stranded. So DNA, it does have several different types of base pairs. So we have adenine, thymine, cytosine, and guanine. So these guys, they will make up the base pairs of the DNA as well as RNA minus T. So thymine is not found in RNA, uracil is. But we'll talk more about that a little bit later on in the lecture. But for DNA, A always pairs with T and G's with C's. So whenever you are going to be like translating or transcribing any sequence of DNA that I may give you, we'll go over an example. But A's go to T's, G's go to C's. Now if you're doing RNA though, it's A goes to U and G's go to C's. So the only difference is that T becomes a U whenever we're talking about RNA. But we're going to first talk about DNA though, and then we'll go into RNA. So each species has a genome with a certain set of genes already. So a gene is a very distinct series of nucleotides. So it's a specific strand of DNA, and that gene will serve a specific function. So every gene, so let's say that a bacteria had a gene for resistance, that gene is specifically for resistance towards penicillin. Or if we're going to talk about like humans, like we have a gene that codes for like eye color. So if you have a gene that codes for blue, then you're going to have blue eyes. So that's the idea behind a gene. So RNA specifically is the product of most genes, and that is called mRNA. So messenger RNA, we're going to talk about him later on. This will specifically direct the synthesis of a specific protein. So from our mRNA, we're telling the cell that we want to make this specific protein, and we'll go into detail about how do we make proteins itself. And then the use of the gene information to make mRNA, and then the protein is called gene expression. So you have your mRNA says to make this protein, and then from that, your gene will be like, okay, this is how I'm supposed to express, meaning am I supposed to be turned on? Am I supposed to be doing this? So gene expression is what does it do? It's like its function. So we're going to go specifically into the genome of bacteria right now. So this looks very scary, but with this side, I really just want you to know the concepts of different parts of the bacterial genome. So we have something called a promoter, so that's this pink region. So this is where the information is determined when a gene is expressed that is contained within a DNA sequence, and that's called a promoter. And then we have something called an operon right next to the promoter, in this case right here. Oh, I'm sorry, operon, not operator. Got confused. So the operon is the promoter with the operator and all the genes in it that are needed to be sequenced. So an operon is a collection of genes that is going to be transcribed on a chromosome into RNA. So what's going to happen is that essentially is that this promoter is going to turn on this operator, and this operator is going to be like, make these genes into mRNA, and then once we have mRNA we can go into protein. So it's kind of like a three-ish step process. But then once we have enough mRNA or enough proteins made from that, what's going to happen is that the regulatory gene is going to be like, oh, we need to stop making all this protein, and it's going to make mRNA, and then this mRNA is going to code for a repressor, and this repressor is going to bind to the operator and it's going to turn it off. So there's going to be no more mRNA transcription, and then no more protein synthesis. So that's the general idea of it. I will post supplementary videos, so definitely look out for those. I'm going to put them underneath modules under supplemental materials. So just in case if you need another different kind of explanation, which is perfectly fine because this stuff is sometimes very hard to grasp, which is okay. Okay, so that's the general idea of how a bacterial genome functions, in the sense of how do we get gene expression to get proteins. So now we're going to go back to some conceptuals. So like I said, normally every bacterium will have their bacterial chromosome, and it's a continuous strand of DNA. It's one big loop, but sometimes they'll have extra chromosomal genetic information, and that consists as a plasmid. So we've already actually talked about this a little bit in class. So a plasmid can either be beneficial. For the most part, it is beneficial to the bacterium, but sometimes it doesn't really serve a purpose and they just uptook it. But whenever they do want to uptake a plasmid, it goes underneath the process called conjugation. We've already talked a little bit about this, but the idea is that what will happen is that that plasmid carries an advantageous gene for that species a lot of times. And what will happen is that you'll have a donor cell that is considered F positive because it has a plasmid. And then you'll have an F negative cell because it doesn't have plasmid. And then what will happen is that they'll come in contact, form a sex pilus, and then what will happen is that this plasmid will undergo replication, and then as it's replicating, it will travel across the sex plasmid into the other F negative cell. And then once that happens, once it's completely replicated, it will recircularize, so become a circle again from the plasmid, and then the sex pilus, they will detach, and now we have two F positive cells. In chapter 9 we go a little bit more into detail about conjugation, but that's the general idea of it. And like I said, an example of that is whenever you have antibiotic resistance, and this is actually how we get antibiotic resistant pathogens. And probably one of the most common ones that you've heard of is MRSA. So that is methicillin-resistant Staphylococcus aureus. So this is specifically how he gets it. And then there's also Microbacterium tuberculosis. So he's also pretty resistant too, and that he gets his resistance the same way through plasmid transfer, so through conjugation. Okay. So now we're going to go into like, how does bacteria actually replicate their genome? So a little background about DNA replication is that it is considered semi-conservative because you always have an original strand, so a parental strand, and then you'll have a newly synthesized strand. So whenever you are doing DNA replication, half of it is always considered old. So that is why it's semi-conservative, which is good because then we're not constantly always making new DNA and having to get rid of it. So it's very advantageous. All species have semi-conservative DNA replication, so that's really nice. And like I said, whenever you're making a new strand, what will happen is that A's will pair with T's and then G's with C's. And then the guy that actually does this, the enzyme that actually copies the DNA, is DNA polymerase. So on the next slide, the next 2 or so slides, I'm going to be naming off a lot of enzymes. Definitely make note of what these enzymes do. So DNA specifically is the enzyme that copies the DNA, okay? So definitely know that. Okie dokie. So the actual process of DNA replication is in 3 steps. We have initiation, elongation, and termination. I definitely want you to know these 3 steps. And these 3 steps are also the same whenever we come to protein synthesis, but in terms of RNA. So it's a little different in what is happening, but it's still the same terms. So for DNA replication specifically, the initiation, this involves whenever our genome is unwound and it's opened, and this is whenever DNA polymerase is loaded onto the DNA. So the enzyme that actually unwinds the DNA and creates this bubble like thing is called DNA helicase. So that's another one. So we have DNA polymerase and we have DNA helicase now. So know those 2. And then during elongation, this is whenever the sequence of DNA is actually happening. So this is whenever we are adding on those base pairs to either side of the strand. And then there's termination. So termination is whenever all replication has within that region is completed. So that means that DNA polymerase has added all the necessary nucleotides, and what is going to happen is that replication is going to stop, and then there's going to be DNA ligase. He's going to come in and tie the DNA back together. So we have DNA polymerase, DNA helicase, and DNA ligase. Now I'm going to show you another image that kind of goes a little bit more into detail. So it's kind of like a little bit more blown up. So as you see that there are 2 different directions of which something is happening, these are the steps of which DNA replication is occurring. So what happens is that the DNA is unwound on the parental side, and what is happening, so we are traveling from right to left in this case. So what is happening is that you unwound the DNA. We have enzymes saying like, okay, we need to replicate this DNA now. And then you have these stabilizing proteins. So this is what keeps it open for the time being until replication has ceased. And then you have something called the leading strand. So this is whenever you're replicating from the 5' to 3' direction. And then you have the lagging strand, which is the other side, which is where you're replicating from the 3 to the 5' direction. So definitely know that. So the reason how you know which side is being replicated is because the strand that's being replicated is facing inward inside this bubble. Okay, it won't ever be replicating on the exterior side. It'll always be interior. So you replicate on the leading strand, so that's the 5' to 3' direction, and then you are also replicating on the same thing but in the different direction from the 3 to 5' direction. Now, whenever you're replicating from the 3 to 5' direction, DNA polymerase does not like that. So what he does is that he does it like in little chunks. As you see, there's like openings right here, whereas on the leading strand it's very continuous. But on the lagging strand, what you're making is something called Owazaki fragments. So DNA ligase, after you do, so after DNA polymerase travels, what happens is that DNA ligase is right behind them to fill in those gaps. So that is also what is happening at the same time on the lagging strand. And then everything will cease once it gets to the point of where, okay, like we're done, but the likelihood is that they're going through the entire DNA, the entire chromosome. But they only do it in small sections. And the reason why they do it in small sections is to minimize error. And then also it's more advantageous to do it that way rather than opening it all up at once, rather than doing it in chunks. So have y'all ever heard of the joke, how do you eat an elephant? You eat it in bites. So it's kind of the same idea with DNA replication. You want to do it in small bites rather than doing it as a whole. So that's the big picture of DNA replication. Now with that, specifically with bacterial, so this is the same thing. They do this exact same process. It's the exact same thing. The only difference is that instead of it being linear, kind of like eukaryotic cells, it's just a giant circle. The main difference that will happen is that replication will actually go in both directions. So in terms of like, it will replicate this way and this way at the same time. So what is happening is that you have that origin of replication that we talked about, so like where that genome is anchored within the bacterium, and then it'll be like, okay, I need to make more of myself, and then the replication bubble will open at the origin, and then it'll just replicate all the way down until you get a whole new one. And then what will happen is that replication will reach on this side and as well as this side and reach that termination point. So once they hit that termination point, then they're done replicating and the cell has finished its process. Now, so with that, I've added some real life scenarios as to why is it important that we know how do they replicate, and that mainly goes with drugs. So there are 2 drugs that I have right here that specifically target bacterial replication. So the first one that I have right here, what it does is that it causes DNA instability. So it kind of breaks the DNA during replication. So remember that we had those anchoring proteins. So the likelihood, I'm not too sure on this, but the likelihood is that the fact that it's causing DNA instability, it's making the DNA less stable, so it's very wobbly, it's not anchored. So with that, it can easily fall apart, kind of like a bridge. So if you don't have support cables on a bridge, it will fall down. So that's the idea. And then I have another one. He does something similar with the DNA, but him specifically, he is a competitive inhibitor of a certain enzyme. And with that enzyme, it is a key, there's a key point of where an enzyme will convert another molecule into something that's called folate. And folate is necessary to make nucleic acids. With this medication, with the sulfamides, he specifically will interfere with making with folate. Sorry guys, total brain fart. So he specifically interferes with the process of making folate, and if the bacterial cell can't make folate, he can't make his nucleic acids. So with the fact that he can't make nucleic acids, he's unable to continue DNA replication. So that's pretty important. So that's just kind of like a real life scenario as to why am I telling you all this shenanigans. So I'm trying to put it into perspective for y'all. Okie dokie. So that was DNA. Now now that we have DNA, now we can go to RNA. So this is the second step, like the second little portion of this lecture. So we talked about DNA, now let's talk about RNA. So, from the DNA, we can get RNA. And whenever we are synthesizing RNA, it's called transcription. So, what we did before is called translation. So, now we're doing transcription. So, the picture below, yes, it is pretty scary. But the reason why it's important for us to know this is because specifically is that if we know how a bacteria codes for its proteins, we can actually create a better fight against them. So specifically in the image that I show here, you see like this red arrow underneath the promoter. That means that the bacteria is actively making these genes. And these genes actually specifically, what they do, they actually code for a protein of an efflux pump. So efflux pumps on bacteria, these guys do exactly what they sound like. They are pumps. So what will happen is that, let's say that you have a bacteria infection, you take antibiotics, but that bacteria has these specific genes listed right here, these MDTs genes, and he has an efflux pump. So what happens is that the bacteria will uptake that antibiotic, but since it has these efflux pumps, it pumps out the antibiotic. So with the fact that the antibiotic is unable to function within inside the cell, you now have a virulent pathogen inside of you and it's harder to treat. So it's definitely important for us to know what proteins are they making and what do these proteins do so we can actually better fight off these pathogens for us. So now we're going to go into like actually how do we make RNA from DNA. So it's kind of similar whenever like we make DNA, there has to be some kind of molecule that's adding on the base pairs. So that's the idea. But what is happening is that you will have RNA polymerase. Instead of DNA polymerase, you have RNA polymerase. So RNA polymerase will come and be scanning the DNA, and then he will reach that promoter region. And from that, the RNA polymerase will be like, oh, the promoter, it's telling me that I need to make mRNA. So what will happen is that RNA polymerase will bind to the promoter region. This sigma factor that you see, this red peanut looking thing, what will happen is that he will pop off, and then the fact that he pops off, RNA polymerase is able to open up the DNA and form like a little bubble. And then what he will do is that he will put his nucleotides onto the DNA and then produce mRNA. So whenever he's producing mRNAs, he's producing AUGs and Cs. So in order for transcription of the DNA to happen, that sigma factor has to pop off. If that sigma factor does not come off, then RNA polymerase cannot unwind the DNA and begin transcribing RNA. So we're actually going to go more into detail, kind of like what we did with DNA replication. So now we're going to do RNA. But before we do that, another side note of why is this important. So there's rifampin. He specifically, what he does actually, this medication, he actually inhibits a certain subunit of RNA polymerase. So what he does is that he will interfere with RNA polymerase, not allowing him to make mRNA for the bacteria. So what he does, instead of interfering with DNA like the other two, rifampin, he specifically interferes with RNA polymerase. So inhibiting transcription. So if you inhibit transcription, you are inhibiting protein synthesis. So it's really good. So that's just another like real life example, like why am I saying all this jargon? Okay. So protein synthesis itself. So I'm going to do a little bit of concepts and then we'll go specifically into what are the steps of it. So we have mRNA, like I said, whenever you are doing, whenever you're transcribing RNA, you are making mRNA. Now, what we need to determine is where do we know when to start making it and when do we determine when to stop making it? So, in order to start making our mRNA, there is a specific sequence that it is looking for to tell it to start. And that sequence specifically is AUG. I'm actually going to type that in. I should have typed it in. So that is AUG. So, a terrible mnemonic to remember what is considered the start codon. So a codon is 3 nucleotides together. So A, U, and G are 3 nucleotides. So in order to remember what is your start codon, so AUG is the abbreviation of August, and school starts in August. So that's one way to remember it. If you don't like it, then that's perfectly fine. It's kind of lame, but that's the best I got. And then there's the stop. So how does a cell know when to stop? Well, there are 3 stop codons. So there's UGA, UAA, and UAG. So I do also have a mnemonic for it, and it's really terrible, and I hope I don't offend anybody. Like, I hope nobody's from Georgia or is an Aggie fan. But this is my terrible mnemonic. So it's U Aggie from Georgia needs to go to AA. So I really hope it's a bad funny, guys. So hopefully nobody is upset with it. But that's kind of a lame way to remember what are the stop and start codons. Okay. Oopsies. So what has to happen before we even start this process is that the bacterial ribosome has to start translating mRNA before we can even complete transcription. So meaning before we can even complete making proteins. So we're gonna go into more detail as to what is the structure of a ribosome in the next few slides. But before we get there, I want to talk about tRNA. So this guy, he is specific for protein synthesis. So we had mRNA, but now we have tRNA. So tRNA, what happens is that he attaches to an amino acid, and it's called a charged tRNA. So essentially what is happening, so you have your, so it's really small, sorry guys, but you have your mRNA right here, and then tRNA, he comes in and he literally puts on like bloop, bloop, he puts on the other codons that are the complement to that strand. So that's what tRNA does. So he will attach the proper amino acid. Okay, so let's practice making our proteins. So on the test, I will expect you to be able to do this. So on the test, what I will most likely do is that I will give you a DNA sequence such as this. So I'll give you something like this on the test. And then from that, what I would want you to do is be able to transcribe it into mRNA. So that means that you will put in something like this. So I'm just going to copy and paste real quick. So you would have something like this. So C's go to G's, A's go to U's, and then C's go to G's, and then T's to A's. So from that, what I would want you to be able to do is use the chart below to determine your mRNA, what proteins, what amino acids are you going to be putting out. So from that, what you need to do is go to your mRNA and find your start codon. So your start codon is AUG, which is right here. So AUG is your start codon. So with that, what needs to happen is that you need to put in the corresponding amino acids. So I'm going to go ahead and actually just copy and paste it, and then I'll talk you through it. So let me get the spacing right. It's going to drive me crazy. Okay. So like I said, AUG is your start codon. So the way that I got that is that first position represents the first letter, and the second position, second letter, third position, third letter. So AUG is our first codon. So we have A, U, and then G. So right here, so that's how I got the met/start. And then our next one was ACU. So A, C, and U, so that's tyrosine, so THR. And then you'll do that for every single one until you reach, until, like, if I only said do the first 4, then you would just start right, you would stop right here. But in actuality, what will happen is that it will keep going on and on and on until you reach a stop codon. And your stop codon in this case is UGA. So you have U, G, and then A. So we have that guy. So then the cell will be like, okay, I made what I needed to make. That's the end of my amino acid chain. So on the test, I definitely want you to be able to do that. I know that was really quick, but in class, I'm sure we're going to do more. And then also, like I said, I'm going to post supplemental videos for this. Okay, so now we're actually going to go into the process of protein synthesis. So just like with DNA replication, it does have 3 phases. It does have the initiation, elongation, and termination phase. But what is actually happening? So ribosome itself, it is a very complex unit. It has two main units. It has the 30S and then the 50S, which is this big guy. So what will happen is that the 50S subunit will join the 30S subunit and make a complete complex. So make a complete ribosome in that case. So whenever that happens, what is happening is that the 30S subunit will get the sign of the start codon, so the AUG, and then that will trigger the 50S subunit to come in and bind to the other subunit. Okay? So if you don't have a start codon, then the ribosome itself cannot become a completed complex. So it has to happen. And what's specific to bacteria, so we don't have this, but one of the signals is that FMET right there, so that formal group, so that is specific to bacteria. Just a little side note though. Okay, so once we have a completed subunit, what will happen is the following. So that is the initiation process of making the complete complex of a ribosome. Now the elongation, the purpose of it is to add amino acids to create a chain. So with that, what happens is that you have your total brain fart. So what you do is that you have this A, so there is an EPA, as you see in the 50X, so that it has 3 sites. So the idea is that what's going to happen is that tRNA is going to come in and add the amino acid to that A part. And then what's going to happen is that a peptide bond is going to form between the A and the P. And then in order for like the ribosome to make room so the A part is free again, what has to happen is that there has to be a shift of the ribosome. So the ribosome will shift So now what was in the P place will go into the E place. What was in the A place will go to the P place. And then whenever that happens, the A site is now empty and can now receive another tRNA. Okay? So, overall, what is happening in 4 simple steps is that you want to add amino acids. So, in order to do that, you add it to the A site. Now, what will happen is that the 50S subunit, so the top subunit, what it will do is that it will form a peptide bond between the amino acid N and the new amino acid that was just added. So there will be a peptide bond between what is in the P site and what is in the A site. And then what will happen is that the peptide will be transferred into the P site in the A site. So they're like that shifting that I talked about. And then what will happen is that after that shift has occurred, the A site is now able to accept another tRNA amino acid. So that is the big picture. The big thing that I might ask from this is like, what is the order of which the sites are used? So like, is it E first, P first, or A first? That probably will be the only thing that I'll ask from this, because it's very nitty gritty, and I just really want you to just know the big picture. And the big picture for this of elongation is what is the order of the sites that are occurring, and then who is actually bringing in the amino acid to add, which is tRNA. Okie dokie. So that was the elongation. Now we have termination. So what will happen is that the peptide bond will terminate once we hit that stop codon. So once that codon comes into the A site, what will happen is it'll transcribe it, it'll finish it off, and then the ribosome subunits, so the 30 and the 50S subunits, they will fall off, and then they'll just be floating around until needed again, and they'll start the process all over again. So once we get that stop codon, the ribosome subunits will fall off of the mRNA, and we will no longer be doing protein synthesis. Okay, we're almost done, guys. Just a few more slides. So this is kind of just like another side note, like, why are we doing this? So it's important that we know how protein synthesis occurs, because then we can have our antibiotics target that specifically. So probably one of the more common antibiotics on this chart that you've heard of is maybe tetracyclines. So these guys, they specifically, what they do is that they block the A site. So they will block the A site of a ribosome. So with that, then there's no protein synthesis occurring, because that A site is always taken up, so you can't add any more amino acids. I think that's all I wanted to really cover on this. So this is, like I said, it's kind of like, why is this important? So mainly the big reason, at least in terms of this lecture, it's important because of drug therapies. Okay. So now we're going to go into another concept of how do we regulate gene expression. So there are some proteins that are always required, such as like RNA polymerase. So RNA polymerase is specifically required for growth. So you always need him. You're always needing to make proteins of some sort. So most proteins are needed only under limited circumstances, meaning that they're not necessary all the time. So they're only needed if you need to metabolize something, and then they're not needed anymore. So how does a cell know when to utilize and when not? And that's what we're going to talk about in terms of bacteria. But before we do that, we're going to go into just some basic categories, and then we're going to dive into one category. So there are several categories of which how gene regulation is done. So there's actual changing of the genetic sequence. So this is whenever microbes, they will change their DNA sequence in order to activate or deactivate a particular gene. So instead of it originally being like TTTs, it'll turn it into CCCs. So with that, then it disables the gene whenever it is turned into mRNA and protein. Control transcription. So this is whenever we talked about this earlier with the operon and the repressor. So this is specifically whenever you state that you're going to make it, but then you're just like, okay, I've made enough, I don't need any more. And we're going to dive more deeply into that one in the coming slides. And then the last two is that there's translational and there's post-translational. So translational, it controls transcription initiation. So this is whenever it recognizes a specific repressor protein. So it will regulate when are we going to do transcription and when are we not. And then there's post-translational. So this is the control of proteins that are already made. So we've already made proteins. Now what we need to do is give those proteins marching orders, essentially. So you'll tell the protein, so this is not accurate, but the idea is that so you make your protein, so you have all your proteins, and you're going to tell them like, okay, from 2 to 3 you're going to be working, but then after that you're not working anymore. So that's the idea of activating and deactivating them. Or if you've already utilized them, they've done their job, what you can also do is degrade them, meaning that you break them down, and then if necessary again, then you'll make them again. But us specifically in this class, we are going to talk about the control of transcription. So with that brings us into the concept of repression and induction. So with induction and repression, it's the idea of like you're inducing transcription and you're repressing transcription and translation. So all I want you to know is the idea, or 3 concepts of each. So with induction, you have the operon is turned on, and then with the fact that the operon is turned on, you're able to do translation and transcription. And then also it operates in a catabolic pathway, whereas repression is the opposite of it. So repression is where you don't have the operon on, so it's off, and then there is no transcription or translation, and it's an anabolic pathway. So that's the idea that I want you to get from induction and repression. That's all I want. The only reason why that's all I want is because you can take a whole class on this, and I just want you to know the big picture of induction and repression. So with that, we're actually going to go into a more specialized pathway for bacteria. So this is kind of like a little side note, but it's an example of a gene in terms of induction and repression. So this is going to be the DTX gene. So this is known as the diphtheria toxin. So if you have diphtheria, then a lot of times diphtheria itself will produce a toxin, and the toxin gene is DTX. So that is whenever you have, oh, I can't even see it. So right here, so the first part is showing you the idea of repression and induction. So what will happen is that if you have high iron levels, it acts as a repressor. So it's a DTXR repressor. So with that, it binds to the regulatory region. So it binds to the operon promoter region, somewhere in there. And then with that, no toxin is being expressed. But whereas if you have low level of irons, what's happening is that the RNA polymerase is able to bind to the promoter region, or to like the regulatory region, and actually allow for transcription, translation to occur. And with that, you have expression of the diphtheria toxin. So you kill you have the potential of killing your host because of that toxin being produced. So this was just a real life example. So this is an example of repression, and this is an example of induction. Now, with the lac operon, I actually have a video for that, but I'm not going to show it because it is a relatively long video. So please um, watch it on your own time. Also, I will be showing it in class, or I should have shown it in class. And then with that, I want you to definitely be able to tell me, what is the lac operon? What does it do? So this is in terms of bacteria. So we don't do this. Only bacteria does it. Once you watch the video, this will make a lot more sense. But the big picture, lac operon is a regulator of a gene. So it's a more specific one. The lac operon is, it's found in E. coli. E. coli contains genes specifically for regulating lactose. So the sugar lactose. So the lac operon has to do with metabolism of lactose. And it is only expressed whenever lactose is present and glucose is not. So that's another thing. Another key factor of the lac operon is that it will, like I said, it regulates in the response of lactose and glucose being present. So there are two things that will regulate whether or not if the operon is turned on or turned off. So there's the lac repressor, and there's also the catabolite activator protein, or CAP. So that's what you see right here. So the CAP protein right here, and then the repressor protein. So with the repressor protein, what it does is that it kind of acts like a sensor, like sensing whether or not if lactose is in the environment. And then what it does is that it normally blocks transcription of the operon. So it'll block it, but it stops acting as a repressor whenever lactose is present. So if lactose is present, then what will happen is expression will happen. So you will get transcription will occur. Okay? And then one other thing, one little side note, and then I'm going to go into dissecting this figure, is that you have a chemical signaling molecule, which is CAMP. So this CAMP right here. So he is a signaling molecule, and he specifically is synthesized from ATP. What it does is that it's exactly what it sounds like. It senses the environment. And what will happen is that whenever there's high levels, CAMP will bind to its repressor protein, so the CRP. He'll bind to that, which in turn will bind other sequences of the DNA. Okay, so now I'm going to go into dissecting that figure. Okay, so the figure below, we are going to dissect it. So with the first part, over here are the contexts of which the environment is. So we're just going to go with the first one. So there's 4 different environments that we're going to talk about. So if we have low glucose and there is lactose available, what will happen, so meaning that we have no glucose but we have lactose, that CAP protein will bind and the repressor will not. So if CAP protein binds, the repressor will not, and with that we will have a strong transcription. Okay, so now the next one is where we have high glucose but no lactose. So with no lactose, that CAP protein will not bind, but the repressor will, because no lactose is available. And with that, genes will not be expressed. Now, what if we have no glucose and no lactose? So what will happen is that the cap will bind and the repressor will bind, and with that, there's no expression of lact genes. Now, what if we have high glucose and high lactose? What will happen is the cap will bind. I'm sorry, cap will not bind. No cap will bind, and then the repressor will not bind. So you see no cap gene, and then there's no repressor gene. So that means that there's going to be little to like no expression of, very minimal expression of lact genes. So on the test, if I were to give you just this part of this figure, I would want you to be able to tell me what are the conditions, whether or not glucose is present or not, as well as lactose. So that is what I would want you to be able to tell me from this figure. Okay, so we're almost done. 2 more slides. Now, what if there is a response to the extracellular environment? So bacteria does sense its environment. It definitely will respond to its environment. And a common mechanism that bacteria use to sense their environment and to transmit information inside the cell is normally a two series component, and that's through protein phosphorylation. So protein phosphorylation is where it will sense something on the outside. So what will happen is that it will have these proteins throughout its membrane, and something will happen, an extracellular or an environmental signal will bind to this protein kinase. And with protein kinase being bound, that triggers a cascade of events. So there is phosphorylation of a protein. So right here, what will happen is that we will have a phosphate group floating around, and then we will have just some kind of regulator also floating around in the cell. And what will happen is that phosphate and this regulator will come together, so meaning that this is phosphorylated. And since a phosphate is attached to a regulator, what happens is that this regulator will bind to the DNA, and with that it will either stimulate or repress certain genes. So something will happen where a gene will be turned on or turned off. So that's just another way of gene regulation. So we talked about intracellular regulation, which was specifically induction and repression and lac operon. And then this is the extracellular, which is the sensor kinase step sequence. Okay, so that was our lecture. I know it was a very hefty lecture. If you have any questions, definitely ask. Definitely look at those supplemental videos that I post. But we did talk about big picture concepts of bacterial genome. We talked about DNA replication, as well as transcription of DNA and RNA, like what are the steps of each. We talked about protein synthesis and the translation process, as well as how do we regulate the gene and the concept of induction, repression, and the lac operon, as well as external signals. Okay, guys, thank you.